Here we discuss the layers of the skin. There are 3 distinct regions that compose the skin: epidermis, dermis, and hypodermis. The epidermis is the upper or topmost region, the most superficial, in that when you look down at your skin, you're looking at the epidermis. It's a very homogeneous layer, relatively thin compared to the dermis. It's composed entirely of one tissue, keratinized, stratified, squamous epithelium, which, thinking back to our tissues, is multiple layers of squamous epithelial cells. However, remember that these cells are really only squamous as we approach the apical or free surface. At the bottom, next to the basement membrane, we're seeing globular, cuboidal, or columnar cells. Also recall that this type of tissue comes in 2 varieties, keratinized and non-keratinized. Keratinized indicates that the tissue undergoes a process called keratinization. Keratinization is a process that fills the cells with a tough, protective protein called keratin that makes them even better at protecting the structures underneath. During keratinization, the cells undergo several changes. Cells are born next to the basement membrane at the bottom of the tissue. And as these new cells divide, they push older cells up more superficially into upper layers. As these cells progress from the bottom to the top, or from the basal to the free surface, the cells are going to fill with keratin, and they're also going to become flatter and flatter or more squamous. Finally, because epithelial tissue is avascular, without blood vessels, the cells also die as they're cut off from the blood vessels in the tissue underneath. Remember, blood supplies the oxygen and the nutrients that the cells here need. So, as they become further and further away from those blood vessels, they're no longer able to sustain life. This creates distinct strata or layers within the stratified squamous epithelium. This whole process takes 4 to 6 weeks for a cell to be born at the basal layer and then be pushed way up to the free surface. When cells divide quicker than this, we have conditions like dandruff or psoriasis, where we see cells being born and then sloughing off within 7 to 10 days. So losing the cell in this amount of time is completely normal. It's a normal part of the maintenance of this tissue. But any quicker and you start to see some issues. Looking really quickly here, this is an image we looked at last time, but you can see these distinct layers where we have the dermis would be under here, basement membrane would be this pink line here, and then our bottommost or deepest layer is this stratum basal, where we have these living cells that are very metabolically active and very mitotically active, always dividing. As they divide, these older cells are pushed up into these more superficial strata until finally they reach this stratum corneum. And at this point, they're totally full of keratin. They are completely dead. You can also see the change in cell shape, right? From kind of a cuboidal, I guess, cell here to a fully squamous cell at the free surface. Now, there are 5 layers, and in 163 lab, we look more closely at those 5 layers. But here in lecture, we just concentrate on the 2 most distinct layers, which are the bottom and the top, the stratum corneum and the stratum basale or basale. The stratum corneum is your most superficial layer or outermost layer of the epidermis. It's usually pretty thick, average 25, but could be 20, 30 layers of dead squamous cells filled with this keratin, this tough fibrous keratin. You may have heard of this word keratin because it's often found in products that treat the integumentary system, like skin care, hair care, nail care products. It's the protein in all of these areas that gives strength and toughness to the cells. Some have more than others, like the nails have more keratin in them than the skin. This is usually the thickest layer, though it varies. And as we spoke about, it can be lost prematurely or quicker than we would like. The stratum corneum is an expendable layer. It's made solely for protection. Again, the cells are completely keratinized. They overlay each other like scales with lipids surrounding the cells that adds more waterproofing and extra protection. And these cells are continually being lost to the environment. We're always losing these cells to the point that we lose about 1 kilogram of dead epithelial cells every year. In response to changes in our environment, the stratum corneum can get thicker. This is what happens when an area experiences more friction. The stratum corneum will thicken to protect against that friction. So we just get more layers here. A corn is the callus, same thing, except it's over a bony protrusion. Now, we're always losing these cells, right? It's a big component of household dust. And unfortunately, one of the many diverse organisms that surrounds us really likes those cells, and that is this house dust mite. The mite is an arachnid, so it's related to spiders and scorpions, and it's usually pretty harmless in general. Mites are detritivores, meaning they gobble up dead, decaying things. But this particular house dust mite, what it eats is dead, shed squamous cells that we lose from our stratum corneum. So it's going to like where we spend the most time, in our houses, in our bedding, in blankets, in carpet, in fabric curtains, in our couches, places like this. And unfortunately, that feces that the house dust mite excretes can worsen or cause household allergies as well as worsen asthma symptoms. So if you find yourself having non-seasonal allergies all year round, it may be because of these mites. There are some things we can do to prevent having a lot of them around. We can keep the humidity low, so a dehumidifier or the AC unit running can drop humidity at or below 50%, which they don't like. And then removing or decreasing carpets, fabric curtains, upholstered furniture, getting rid of dust with a damp rag or mop. If you use a dry rag, it just stirs it up, which will increase allergy symptoms. And then washing bedding and blankets once a week in hot water, 130 to 140 degrees Fahrenheit. And while we can't completely eliminate them, we can decrease their numbers and decrease our symptoms. They are quite small, so you can see that these are squamous cells. So if a cell is that big, well, the mite isn't much bigger than that. So that's our stratum corneum way up at the top. The stratum basale is at the bottom, next to the basement membrane, so the deepest layer of the epidermis. And it's exactly the opposite. So instead of many layers of squamous cells, we just have one layer of cuboidal or columnar cells that are firmly attached to the dermis with hemidesmosomes and firmly attached to each other with desmosomes. While the cells of the stratum corneum are dead, the cells here are very much alive, and it's mitosis here that allows cells to be pushed up into the other layers. So here's another image, a diagram. This is how they appear in the skin in real life. When we look at these layers, sometimes people mix them up with different types of tissue. And I just want to caution you against that. This whole thing, the epidermis, is one tissue. It's our stratified squamous epithelium. These different layers are just different regions, like a layer cake, different layers of that one type of tissue. It's this process of keratinization that makes them look different. The underlying dermis is quite a bit thicker, and it's quite heterogeneous. So while the epidermis was all that epithelial tissue, here we're going to have a lot more going on. The bedrock, the base, is going to be dense collagenous connective tissue. And if we want to get technical, it's irregular collagenous connective tissue, packed dense with collagen fibers that are going in multiple directions to give us strength and resistance to stretching and tearing in multiple directions. To give you an idea, if you are familiar with leather, that is the dermis of a cow or another animal. So you can kind of get a feel for how tough this tissue is while still maintaining a lot of flexibility and ability to move. Here we'll find fibroblasts to secrete and maintain those fibers, a few adipocytes in the mix, macrophages for immunity, and then also nerves and receptors, sensory receptors, some smooth muscle, some glands of different types, both blood and lymphatic vessels, and then also hair held in hair follicles. So there's a lot of different structures, organs found here. While this is irregular collagenous connective tissue, the fibers are oriented in certain directions more than others, and this gives us cleavage lines. These are areas where the skin is more resistant to stretching and tearing, just due to more fibers being oriented that way. This is important for scarring during surgery, because cutting with these cleavage lines in the same direction will result in much less scarring, because it disrupts fewer fibers. Overstretching these collagen fibers leads to stretch marks. It's a form of internal scarring. Here we can see those cleavage lines. So if we disrupt more of them with an incision, that's going to just lead to more fibers being disrupted, more time needed for healing, and then more scar tissue when that healing occurs versus going with the collagen fibers. The dermis can be divided into two areas. The papillary layer is a thin layer of more loosely organized more areolar connective tissue that lies right underneath the epidermis. So this is touching the epidermis. It's called the papillary layer because it is composed of dermal papillae, which are wave-like projections that extend up into the epidermis. Each projection contains blood vessel loops or capillary loops and sensory receptors. The blood vessels supply nutrients and oxygen, remove waste, as well as regulate body temperature by either dilating or constricting, widening to increase blood flow or tightening to reduce it. The sensory receptors packed in here are going to increase sensitivity. And essentially, the projections increase the amount of contact and the amount of area for these vessels and receptors. We can see an extension of these when we look at the palms of our hands and the soles of our feet. The shape of fingerprints and footprints is the epidermis that is laying over these dermal papillae. The dermal papillae are going to push up the epidermis in these wave-like patterns, which we can see as fingerprints and footprints. These have a function as well, as they work to increase friction, increase grip in these areas. Most of the dermis is the reticular layer, so about the 80% of the dermis, the majority. Here we can actually also see papillary layer, so right underneath the epidermis is the papillary layer, and everything else is the reticular layer. It goes down much deeper than this. This is just a small part of it. This is where the other accessory structures and organs of the dermis are found. So hair follicles, sweat glands, oil glands, and blood vessels, nervous tissue as well. So here is the diagram from the book that shows epidermis versus dermis. We can get a feel for the thickness of each. We can also see how the epidermis is composed of just one type of tissue versus a lot of structures found in the dermis. We can see that the base has these collagen fibers, okay, going in multiple directions for our dense, irregular, collagenous connective tissue. We can also see the papillary layer with these capillary loops, and then most of this is reticular layer. Now, quickly before we move down, we have skin color. Right? This is determined and influenced by pigments in the skin. The major player here is melanin, which isn't just one pigment. It refers to a group of pigments that are chiefly responsible for skin, hair, and eye color. There is another pigment, well, a couple other pigments that play a role. Keratin is a yellow pigment that can be found in the skin, but it's found first in plants like carrots, sweet potato, squash, pumpkin, red bell pepper. Can make these plants look not just yellow, but orange or red. Also, blood circulation through the skin can play a role. Blood is red, so due to increased or decreased circulation, that can add a pigment. And then the thickness of the stratum corneum plays a part too. So melanin here, it's a group of pigments, mostly brown to black, though there are yellow and reddish pigments as well, again responsible for most of our pigment. Melanin is important because it provides protection against UV light from the sun. So it absorbs this light and acts as a natural sunscreen for us. It's produced by special cells called melanocytes. Produced in the cell, then packaged into vesicles, which move out into the processes or projections of the cell. And then the epithelial cells nearby will eat up these processes. So gobble up the melanin. And in this way, all of the cells of the epidermis will be protected by this pigment. This image from your book shows how this is done. So these are the normal epithelial cells all around, and then our melanocyte here with the little vesicles of melanin called melanosomes, because of course there's a special name for them. And then all the neighbors here, all our normal epithelial cells can take up bits of these processes and ingest this melanin, which will then work to protect. It'll move to make a protective veil over the nucleus and protect the DNA of the nucleus against the damage that can occur when UV exposure happens. Freckles and moles are just accumulations of melanin where there's a lot of melanin found. The amount of melanin produced is a factor of genetics, exposure to UV light, as well as hormones like estrogen. It's genetic factors that are responsible for how much melanin is produced and thus the differences in skin color between individuals. We all have the same number of melanocytes roughly, but the amount of melanin, the types, different arrays of pigments, and then the distribution of melanin is going to vary. Albinism is the complete absence of melanin due to just a single base mutation that makes it so an individual can't produce melanin, or a protein that's necessary for producing melanin. So people who have albinism are extremely susceptible to skin cancer and have to be very careful about going out in the sun. So melanin is really by and far the most important pigment for determining skin color. Keratin is it's a yellow, orange, maybe slightly reddish pigment. But again, it's found in plants. It can influence our pigment because even though we don't produce it, it can accumulate in the skin. It accumulates in the lipids of the stratum corneum, those waterproofing lipids, and also in the adipocytes or fat cells of the dermis and hypodermis. We ingest it as a source of vitamin A. So it's a precursor. You may have heard of beta carotene, precursor to vitamin A that our bodies then turn into usable vitamin A. And when we have too much, it's toxic. So instead of converting it to vitamin A then, we will convert excess, or rather store excess, in these areas of the body. Thus can give individuals an orangish hue. It's not toxic in this way. So this is a way for the body to protect against the toxicity of excess vitamin A. It looks scary, but the only thing that you need to do in this case is stop eating orange plants. I've only heard of this in the context of toddlers undergoing food jags, where they really, really like to eat one or a few things. So say your kid just wants to eat carrots morning, noon, and night, and you take him to the doctor. You haven't seen anything wrong because this has happened slowly, but the doctor hasn't seen your kid for a year. And she looks at him and says, is your kid looking a little orange? And just stay off the carrots and everything will be fine. Now, this is also not a big player, but hemoglobin. This is the protein found in red blood cells that gives them, and blood, its red color. It's responsible for binding to oxygen. So it's what gives red blood cells their function. So due to increased or decreased blood flow then, we may see hemoglobin playing a small part in influencing pigmentation. Now, how melanocytes work is that UV light stimulates them to produce melanin. So the more UV light they are exposed to, the more melanin they will produce as a response. And then that melanin is going to build up in the skin and protect against that harmful UV or ultraviolet radiation in sunlight. They don't have an infinite capacity though. So when we exhaust their ability to produce that melanin, a person can experience a sunburn. So that's the skin reacting to that UV exposure. Long-term exposure to sunlight, lots of sunlight, can result in more long-term damage. So it not only can cause this burn of the epithelial tissue in the short term, it can cause the elastic fibers to become less elastic, to clump up and become stretchy and more leathery, so cause structural changes in the skin. And most dangerously, UV light alters the DNA in cells. It causes it to mutate. A mutation is a change, so it just causes the DNA to change. And this can be a cancerous change, resulting in skin cancer, which we'll talk about later on. Because the skin is so exposed, it can be a useful diagnostic tool. It can be assessed very quickly and without having to rely on difficult or invasive techniques. Redness is an indicator of increased blood flow. It can be a sign of fever, hypertension or high blood pressure, inflammation, an inflammatory response, or allergies. Pallor is the opposite, paleness. It's an indicator of anemia, low iron content, or low blood pressure or hypotension. Cyanosis is a bluish tinge, so not neon blue or turquoise, but just a slight bluish, pale bluish color that's a result of decreased blood oxygen levels. Jaundice is one that you may have heard of. This is a buildup of bilirubin, which has a yellowish color to it. Bilirubin is normally processed by the liver, but if the liver is compromised or just unable to handle that bilirubin, it is stored within the skin, resulting in a yellowing of the skin, which we call jaundice. So it's a sign of a compromised liver, whether that's a baby whose liver has not quite developed yet, or something like cirrhosis or liver cancer. Much less common is bronzing, which is a sign of an very rare endocrine disorder called Addison's disease. And then finally, bruising, right? Bruising is broken blood vessels that have allowed blood to leak into surrounding tissues, changing the color of the surrounding tissue, and that can certainly be seen in the skin as well.